CMAP stands for Comparative Map Viewer. And what CMAP allows you to do is compare one map to another. In this case, what we're looking at is a comparison between linkage group C2 in the map set GM Composite 2003 versus the linkage group C2 in the map set GM Consensus 4.0. We could easily change this to other maps and compare, say, the C2 linkage group from GM Composite 2003 to that of any number of other map sets and linkage groups that contain at least one marker in common. So why would we want to do that? Well, if we were to instead compare the linkage group C2 in Composite 2003 to other homologous chromosomes to C2, we might be able to look for commonalities in the QTLs, which might also indicate the areas that are homologous, and eventually we might be able to turn that into a list of putative candidate genes being on both of the chromosomes in the genetic area responsible for the same trait. So how do we change maps? Pretty easy in CMAP. We scroll down to the Map Options dialog, and the first thing we want to do is to delete the GM Consensus 4.0 C2 map. And we do that by clicking the delete set. And CMAP redraws the maps, and as you can see, it's only displaying C2 from GM Composite 2003. So we scroll back down to the Map Options dialog, and this time we want to add a map. Now we can add maps to the left side of the reference or to the right. In this case, we'll add a map back to the left side. And we'll do that by clicking on this link. We click on this link and it opens up another dialog which allows us to select what map sets we want to pick the linkage groups from. So if we pull down the menu bar, we can see that we're presented with a whole list of maps that have at least one marker in common with the Composite 2003 linkage group C2. But we want to look at homologous regions and other chromosomes within the GM Composite 2003 map set. And so when we highlight that, we notice that there are 16 chromosomes that have at least one marker in common to C2. So we'll pick the GM Composite 2003 map set, and then you can see in the dialog box that a number of other linkage groups have at least one marker in common, and they're listed in here, and we can scroll down and see that indeed there's a list of about 16 chromosomes, all having varying numbers of markers in common. And if we wanted to, we could add all of them by simply clicking the all. But in this case, we'll just add back linkage group A1. So we'll select it and then click Add Maps, and CMAP will redraw the map with, this time, the comparison between A1 and C2 of the Composite 2003 map set. And you're presented with the usual CMAP type of display where markers in common are in red, and they're also connected by the blue lines. And this display, if we zoomed in, we could try to find areas that have the same type of QTL in both chromosomes, indicating a homologous region in both of the chromosomes. This has been a SoyBase production. For more information about SoyBase, go to www.soybase.org.